Hi, I'm Harihara Subrahmaniam. I'm a graduate student with the Department of Computer Science, advised by Dr. Mihai Pop at the University of Maryland. I thank the organizers for giving me an opportunity to present my research. Today, I'm presenting a pipeline, Pyrate, aimed at identifying novel phage elements from assembly graph variants. I would like to begin my presentation with why the study of phages is important. The discovery of antibiotics in the 20th century is an important milestone in the history of modern medicine. While antibiotics remain indispensable to several areas of science, pathogens evolved rapidly and counter the drugs designed to kill them. In the bar chart, we see more than 200,000 people are infected by C. difficile in the US alone, and more than 23,000 people die annually. It is also conjectured that more people will die due to a superbug infection over cancer by 2050. Bacteriophages are seen as effective complements to antibiotics. Bacteriophages are viruses that affect bacteria and eventually kill them. There are several advantages of phages over antibiotics. First, it is host-specific. Because it is host-specific, it wouldn't harm other useful species of the bacteria in the community. Second, phages are self-replicating. Hence, the phage is there as long as the target bacterium is alive. Third, phages can degrade biofilms, which significantly degrades the efficacy of antibiotics. Hence, the discovery of new phages is crucial to drive therapies forward. Let me explain why I think metagenomic assembly graphs are the place to look for novel phage insertions. Let's begin by examining the issue with the existing phage classifier methods. The existing methods are heavily reference dependent. Most of the known phage databases are sparse. Since phages mutate very quickly, it's hard to characterize them just based on references. Second, with the decreased cost of genome sequencing, metagenomic sequencing is becoming more and more ubiquitous. This is well illustrated by the growth in the size of the sequence read archive database. Finally, I would like to talk about GRASphage. GRASphage is the first computationally identified phage genome. To identify the 97 KB GRASphage genome, all sequences that matched previously published viral microbial databases were removed, and the remaining sequences were cross-assembled. Our lab is interested in computationally detecting phage like GRASphage from metagenome sequencing data, and we think that our tools that detect structural variants in assembly graphs can provide novel approaches to this problem. Let me begin with describing an assembly graph. Assembly graphs are directed, and the nodes represent contigs or unitigs, and the edge represents mate pair support. To this end, our lab developed a tool, Metacobble, that detects structural variants from assembly graphs while scaffolding contigs. For the study of phages, we are interested in two kinds of structural variants, namely the indel bubble and the interspersed repeats. Indel bubbles are triangular regions in the assembly graph. Indel nodes could represent specialist phages. Specialist phages are those that affect a specific species in the microbial community. Interspersed repeats are a node with a signature shown in the green bubble. Interspersed repeat nodes tend to have a high betweenus centrality, and these could represent genderless phages. Genderless phages are those phages that could affect several species of the microbial community. Hence, these show up as a node central to different species in the assembly graph. To validate the claim, we try to look at the regions where most of the GRASphage genomes were present. As mentioned earlier, GRASphage was the first computationally assembled phage. GRASphage was generously found in stool samples. Most of the contigs that matched GRASphage genome were found to be interspersed repeats or indel bubbles. In the right, I show an example of an interspersed repeat. Here we see a GRASphage insertion common to two Bacteriaceae species. Having shown that indel and interspersed repeats are rich in phages, we are interested in mining novel phages. To hunt for novel phages, we gather all the contigs belonging to either interspersed repeats or indel nodes from 208 HMP stool samples. Since we are interested only in novel segments, we would want to get rid of any contig that is already found in reference databases. To that end, we blasted all the contigs against the non-redundant database. We scored the contigs based on the breadth of coverage. The rationale behind this step being, If a contig is not very common, the breadth of coverage will be low. We removed contigs with high breadth of coverage. We also removed contigs smaller than 3000 base pairs from further analysis. At the end of the step, we were left with 78,947 candidate phage sequences. We want to look for similar sequences across samples. We do this to see if we can detect similar novel genomic regions across environments. We used MASH, which is a min-hash base distance obtained by sketching k-mers from the sequences to cluster these contigs. Once we computed the pairwise MASH distance between the sequences, we clustered them using the complete linkage method. To identify clusters, we used the cophonetic distance metric. We spotted an e-point in the cophonetic distance versus number of clusters at 725. Of the 725 clusters, 624 clusters were not singleton. We considered these 624 clusters for further analysis. To validate these clusters, we obtained the proteins from the UniProt database using the search keywords phage and integrase genes. To narrow down to clusters that could contain novel phage elements, we did a blastex of these proteins onto the contigs. Of the 624 clusters, we were able to identify 99 clusters with about 1,500 known phage or prophage genes. The figure on the right parameterizes the number of clusters for a given number of samples. The clusters belonging to the left of the histogram essentially signifies that it contains sequences from fewer samples. Hence, these could represent phage sequences that are specific to an environment. As we move along the x-axis, the cluster contains sequences from multiple samples. These could be phage insertions that is more prevalent across the environment. However, the absence of known phage-related proteins in the other 525 clusters doesn't rule out the presence of novel phages in them. This could simply be an artifact of the sparsity in the database. I present a closer view at one of the clusters that contained known phage or prophage-like genes. The cluster 50 had good hits for the integrase genes, inversion proteins, and the recombinase proteins. We also see that this cluster 50 had sequences from over 100 samples, making it common across the population. In this work, we designed a pipeline to mine novel phage sequences from metagenomic assembly graphs using graph signatures, namely the interspersed repeats and indel bubbles. We were also able to establish the prevalence of phage-like genomic insertions across samples. In the future, we are interested in exploring the context to complete the putative phage genome. We also would like to explore sequence signatures corresponding to phage elements in addition to graphic. Thank you.